going to have exam number 3 this week, and I shifted the start date. Okay, so we are going to have it available Thursday till Friday. Okay, and that test or quiz, or sorry, exam number 3 cover 3 chapters, 9, 10, and 12. You are not responsible for chapter 11. 10 and 9 and 10 are very, very linked, as you know, because one of them is talk about the cellular respiration, and chapter 10 is photosynthesis, and the two chapters are connected very well together. So my plan again is to go through test number 2 first and see some of the questions that most of the students didn't get right. And then we can go to chapter 9 and 10 if you have any question for me before the test. So before I start, do you have any question first for me?
You said that the exam 3 is going to be 9, 10, and 12.
Yes.
Okay. And you changed that to Thursday, so that won't be available Thursday till 8 a.m.
Yes, it will be available Thursday till Friday, midnight Friday. Yes.
Okay, thank you.
Yeah, and this will be 3 chapters, and that will leave one exam, which is exam number 4, and that exam covers chapter. It will be 14, 16, and 17, and that will be it. Then we're going to have our final, and the final exam will be kind of cumulative. And I will send an announcement, like soon. So what kind of chapters do you need to focus on for the final?
Thank you.
Okay, great. So let me share my screen with you and go to test number 2. There's few questions that I'd like to go through and cover with you. Okay, can you see my screen?
Yes.
Okay, great. So that one was kind of a little bit harder from if you compare it with test number 1. And I can tell that chapter 5, which is macromolecules, was kind of most of the questions come from that chapter. Student couldn't get it right, and this is why I'm following up with the extra credit, like for 5 extra points that will be added to test number 2, and those mainly from chapter 5. So if you focus more on chapter 5, you will be able to get those 5 points easily, I believe. But I will go through those one more time, okay? Because you're going to see those questions again in your extra credit. Okay, so let me go through some of those questions here. Let me see. Yeah, here, number 1. This was chapter 5. And here it says scanning electron micrograph. Okay, SEM, use a beam of electrons that is transmitted. This is a false statement. Why? Scanning means the scanner. It's scanning the object. So it uses electron beams to scan the surface of the object that you would like to see, okay? If the electron beams are going to be transmitted, then the type of the microscope will be transmitted electron micrograph, or TEM. So the first half of the sentence was right. Scanning electron microscopy uses beam of electron, yes. But this is that is transmitted. This is the wrong here. This is the problem. So that was why most of the students didn't get it right. That was kind of a tricky question. So when you have those kind of questions, I tried very much to read it quite well before you select the answer. Okay. So this is why you need to know this. A scanner or a scanning is scanning the surface. A transmission electron microscopy is basically send electron beams through the cell that you'd like to see. So you can see internal structure of the cell. Scanning, on the other hand, you see only the surface of the cell, okay? That was one of the question here that was kind of tricky. Yeah, here, another one, I can see like almost 50-50, was kind of confused. It says, which of the following is not true about enzyme? Not true. So you need to see which sentence here that doesn't make sense if you talk about enzymes, okay? So first of all, let's go through this. They are usually made of amino acids. This is a true statement. This is a true statement. Enzymes are proteins that are mainly made of amino acids. They lower the activation energy of a chemical reaction. Yes, they are. This is a true statement too. Enzymes, their main job is basically to lower the activation energy of any chemical reaction. Any chemical reaction, before it starts, it needs to be activated. All the molecules need to have some kind of energy to be activated, to be involved in a reaction. Enzyme, the job of the enzyme is to lower this energy needed, okay? Each one is specific to a particular substrate. Also, this is a true statement, okay? So which one is not true? The delta G. Enzyme has nothing to do with the delta G of a reaction. The delta G is a property of the free energy available in the reactant and products. Enzyme cannot mess with this. So enzymes have no correlation with enzyme activity with the delta G of the reaction, okay? So that was the one which is not true, okay? Go further here. Potential energy. What is a potential energy means, first of all? Potential energy, this is the kind of energy which is basically stored in the chemical bond in organic molecules, okay? And they are basically there ready for a reaction to be used during cell respiration, for example, okay? So if you compare those four different molecules or macromolecules, so if you look at the cellular respiration, you have one molecule of glucose. If you break this down during the process of cellular respiration, you are going to generate approximately 36 to 38 molecule of ATP. So now if I compare glucose with ATP, which one of those has more potential energy? Is it glucose or ATP? Definitely it is glucose, because this sugar, you can break it down and release the energy, and that energy can be used to make approximately 36 to 38 molecules of ATP. And we know for sure that the potential energy of ATP is more than ADP. So we can say ADP has more potential energy or more energy than ATP. If you look at glucose and sucrose, this is a monosaccharide. Sucrose is a disaccharide made of glucose and fructose. So generally, when you look at this, you say that sucrose has more potential energy because it has more two molecules. One of them is glucose and the other one is fructose. And each one of those can be breaking down and give lots of ATP. So if you compare the potential energy for those, sucrose comes first, followed by glucose, followed by ATP, and then ADP final. Okay, this is important. So maybe you will see something like this in your final too. Okay, so if you have any questions, stop me please at any time and ask. Any question for me so far?
No from me.
Okay, anybody?
No.
Okay, good. So let's continue here. It says how many molecules of water are released during the polymerization of a 20 monomer long cellulose molecule. Okay, you know that each time when you make polymers out of monomers, so each time there is a linkage between two monomers, one molecule of water is coming out. We call this a dehydration reaction, okay? Let me show you here, share with you another screen. Okay. Okay, can you see this slide here in this presentation?
Yes.
This is from chapter 5. And here is what I'm talking about. I'm talking about here in this is a very good example here. You have each circle here represent a molecule. So you have 3 molecules. They are linked together via covalent bonds. And then if it happens and you need to add another one, so you are making polymer of 4 molecules, here where 1 molecule of water is coming out, okay? So each time there is 2 monomer linked, 1 water molecule is coming out. So this is a very nice equation here. N minus 1, it will give you the number of water molecules needed to make a polymer made of N molecules, okay? So N here is basically a polymer, and the number of monomer in this polymer is N. If you subtract from this 1, that will give you how many water will come out from during this reaction, okay? If you look here, 1, 2, 3, 4, this is 4 monomers, and those are linked together to make a polymer of 4 molecules. So if I ask you how many water molecules came out from this, it will be 4 minus 1, and that will give you 3 molecules of water. 3 water molecules will come out. Okay, between 1 and 2, 1 molecule of water will come out. 2 and 3, another molecule. 3 and 4, another molecule. So together there will be 3. Okay. Do you have any question for this before I proceed?
No.
Yeah, you can stop me anytime if I'm talking like with a high speed, you know. So let me stop share here and go back to the test. So when I say how many molecules of water are released during the polymerization of 20 monomer long molecules, so that will be 20 minus 1, it will be 19. So the right answer is going to be 19 molecules of water should come out when you make a polymer of 20 molecules to make a cellulose, okay? Here, yeah, that was challenging one. The majority of the class got that one wrong. So it says here, the molecular formula of glucose. It gives you the molecular formula of 1 molecule of glucose, as you can tell. And it says what would be the molecular formula for a molecule made by linking 3 glucose molecules together. So if you have 3 of those linked together, okay, so what will be the molecular formula for the end product? So let me go again and show you this. Okay, let me share with you. Okay, so this is here the question. So first of all, you have the molecular formula of 1 molecule of glucose, C6H12O6. So 3 molecules of this will give you the molecular formula to be like that, okay? It will be, you multiply the 3 times every single atom in the molecule. So that will be the molecular formula for 3 molecules of glucose linked together. But in order for those to be linked together and make a polymer, water molecules will come out. So how many? You have 3 molecules of glucose, so 2 molecules of water will come out from this. 2 molecules of water, meaning 4 hydrogen and 2 oxygen. So you need to subtract those from this molecule, and that will give you the molecular formula of the 3 glucose molecule when they bind together or link together to make a polymer of 3 sugar. So a question here to be able to solve it, 2 things you need to do. First of all, you need to get the molecular formula of the 3 molecules together. And then you know that when you make those 3, you need to take 2 molecules of water out. So you'll do a simple math, and you are going to subtract those 2 molecules of water from the molecule, and that will give you the answer here. Carbon is going to be the same because we're not removing any carbon. So carbon is 18, 18. Here hydrogen 36, so we're going to take out 4 hydrogen. See, 4 hydrogen. So that will be 32. Oxygen, 18 atoms of oxygen. You're going to take 2 oxygen atoms, okay? So that will be 16. And that will be the molecular formula of this molecule.
I have a question.
Yes.
Okay, so I'm a little confused. How do you know to take 4 of the hydrogens out to get 32?
Yeah, okay, here. How many molecules you need to link together, first of all? 3. 3,
Yeah.
right? So when you link 3 together, how many molecules you are going to take out based on this equation, where N is the number of molecules. You have 3 molecules of glucose, so how many molecules of water will be taking out? You subtract 1, right?
Right.
So that will give you 2 molecules should come out, right? 2 molecules here.
Right.
So those 2 molecules meaning what? When you say 2 H2O, how many hydrogen in 2 molecules of water?
Okay, so 4.
4. How many oxygen in 2 molecules of water?
2.
Perfect. So if you subtract 4 from this, it will give you 32. If you
Gotcha.
subtract 2 from this, it will give you 16.
Okay.
So in order to solve this, you have 2 things. First of all, you need to know how many molecules will come out, and then do the simple math. So this is always very important equation to know. If I give you, I said, you have a macromolecule or a polymer made of 1,000 glucose unit. How many molecules of water should come out for this molecule, macromolecule to form?
Is it 1?
Again, you have a molecule of, say, starch, which is made of 1,000 glucose, okay? So in order to be able to form this macromolecule, how many molecules of water you need to take out?
I don't know.
Yeah, again, the equation is N minus 1. So N is the number of the glucose subunit. You subtract 1. So 1,000 minus 1 is?
999.
Yes. So this is the amount of water, that molecule that you need to take out when you form 1,000 molecule of glucose.
Okay.
Because each time you are linked 2 sugar together, you are taking 1 molecule of water out.
Okay.
Each time you are linking 2 molecules, you are taking 1 molecule out, okay? So if you are trying to link 1,000 molecule, so you are taking out 999 water molecules to be able to form a polymer of 1,000 glucose subunit,
Okay.
okay? So any question for this before we proceed?
Not from me.
No.
Okay, so let me go back and share the screen again with this one here. So now you know how to solve this. Here another one that was kind of confusing again, because I can see most of the students got that one wrong as well. Okay, what is the difference between an aldose and a ketose? So anything end with the suffix os, ose, os, it's a sugar, okay? So both are sugar, both are sugar. But what is the main difference between aldosis and ketosis is the position of the carbonyl group. And let me show you here, you have a nice slide in your presentation that show you this. Let me share again. Can you see this slide here?
Yes.
And this is aldose and this is ketose, okay? So the main difference, as you can tell, this C double bond O is, we call this carbonyl group, carbonyl group. If you have the carbonyl group at the terminal or end of the molecule, this is an aldose, okay? Because the whole thing here, CHO, is an aldehyde group, and we call this an aldose. If the carbonyl group is hidden in the middle of the molecule, it is not at the periphery of the molecule, like in this case, we call this ketose. So it is not the number of hydrogen or oxygen or the position of the hydroxyl group or anything like that. Because if you look at those two molecules that are isomer, okay? And those are very nice isomer here because they have same number of carbon atoms. They have same number of hydrogen. They have same number of oxygen. Even the position of the hydroxyl group in the molecules are the same. So what is the main difference is the position of the carbonyl group. Carbonyl group here in the middle is a ketose, and if it is at the periphery, at the end, at the terminal part of the molecule, it is an aldose, okay? So remember, this is very important. Little things like this, you need to take care of because you are losing points, unnecessarily. Let me go back. So again, the difference here is going to be the position of the carbonyl group, okay? This one also, opinions of the students was kind of different. So we have like almost less than 50% got it right and more than 50% got it wrong. So when it say which amino acid would most likely be present in the outer side of a transmembrane domain, you need to think about a protein which is completely integrated in a membrane, in the plasma membrane of a cell, and that protein is like a channel. So there are two phases for the protein. One phase is going to basically meet with the hydrophobic tail of the plasma membrane, and one part will be hidden. So you need to expect that the part of the channel of these amino acids that are facing or interacting with the plasma membrane, the hydrophobic part of the plasma membrane or the fatty acids, it should be hydrophobic amino acid, okay? Because you will not see any charge or polar amino acid in this kind of environment. This environment is highly non-polar, highly hydrophobic. So amino acids should be hydrophobic as well. Okay, it makes sense?
I got this question wrong, but I think the only reason I struggled so much is just because the wording. I don't know what it was about that question. It really stumped me.
So yeah, like which one of these was not clear to you?
It's just when it gets to the part where it says the side of a transmembrane domain of an integral membrane protein. That's just that one phrase. I couldn't decipher.
Yeah, if you look at this one, you remember when we in the membrane unit, it talks about the plasma membrane, and the plasma membrane is not made only of phospholipids, although phospholipids are the majority, but there are also proteins in the process involved. And those proteins can be completely integrated in the membrane or can be peripheral. Peripheral meaning at the edge of the plasma membrane, either facing inside, facing the cytoplasm, or facing outside the extracellular environment, okay? So when you say integral, meaning that the whole protein is spanning the whole membrane, okay? It's completely embedded. When you have a protein integrated in the membrane like that, so you expect that when the protein makes the three-dimensional shape, some of the amino acids will be facing the hydrophobic tail, and those amino acids should be hydrophobic. Some of the amino acid side chain will be internal, and those are away from the hydrophobic tail, and those will be polar amino acids. This is the main idea of this.
Thank you.
Yeah, no problem. Okay, let me continue here. I will go to this one later about the delta G and stuff, the figure, as a matter of fact. I will go to this later, but let me continue. Here, again, same deal. Again, macromolecule chapter 5. When I say tripeptide has 3 amino acids. If you talk about a protein made of 3 amino acids, okay? So those amino acids are linked together via tripeptide bond, okay? So a tripeptide here, so a tripeptide meaning a protein or a polypeptide made of 3 amino acids. When you have those 3 amino acids linked together, okay, to make a tripeptide. So how many bond will connect the 3 amino acids?
2.
Exactly. So it's not going to be 3. It is a tripeptide, meaning it's a polypeptide chain, short polypeptide chain made of 3 amino acids. Yes, but the number of bond or covalent bond that link those amino acids are going to be 2. Same like when we mentioned about sugar. So you always have 2 molecules of water are coming out. And when you have 2 molecules of water are coming out, you are making 2 polypeptide, so 2 peptide bond between the amino acids. Okay, so it is not 3. Because when the student read tripeptide, 3 amino acids, they said, yes, it is made of 3 polypeptide bond. No, that should be 2 peptide bond. This is why majority got it wrong, okay? Here, it's a true statement about protein is that when you do denaturation, that will disturb the bond. That makes sense. And the molecule turn into liquid. Take an example of this. If you are, I give you 2 examples. One of them is egg. If you take an egg before you boil it, you crack it, then you will see the yellow part and the white part. The white part is basically albumin, which is protein. It is liquid. And this is folded correctly. It's properly folded. If you take this and you put it in boiling water, when you try to do boiled egg, what is happening? You are basically breaking down and denature the albumin, the protein. So the protein is going to be solid. Okay, so it is the opposite. Okay, another example, milk. In the process of making yogurt or cheese, milk has protein casein, which is liquid. And when you try to add like some bacteria that ferment the sugar, lactose, and make lactic acid, so the pH of the milk is going to drop. And that acidity will cause denaturation. And that denaturation will make casein insoluble. So you will see it like particulated, okay? So the opposite is correct, but not that one, okay? So this is a false statement, okay? That makes sense?
Yes.
Okay, great. When I say the relation between amino acid and polypeptide is similar to the relation between nucleotides and nucleic acid. Makes sense. The building block of proteins is amino acids, and the building blocks of nucleic acid is nucleotides. So that is a very good correlation here. So that's a true statement as well. Here, when also almost 50-50, purine is the measured difference between RNA and DNA. It's wrong. This is false. Because purine, what is purine? When I ask you purines, what are purines? Generally speaking, again, when we talk about macromolecules, this is again chapter 5. Nucleic acids made of nucleotides, right? So what are purines? Any clue? Hello? Any clue?
I don't know the answer to that, sorry.
That's okay. Anybody
I don't
else?
either.
Yeah, so if you talk about nucleic acid or nucleotides made of 3 major parts. One of them is the sugar, okay, which is ribose. In case of RNA, it is ribose. In case of DNA, it is deoxyribose, meaning there is an oxygen minus 1 oxygen atom. And it has phosphate, PO4 phosphate group, and it has a nitrogen base. And the nitrogen base can be purines like adenine, guanine, or pyrimidines like thymine, cytosine, uracil. Okay, so if you look at the RNA and DNA, they have the purines are the same. Adenine, guanine, both RNA and DNA, they have adenine, guanine. Okay, what is the difference then? The difference is the sugar. In case of RNA, the sugar is ribose. In case of DNA, it is deoxyribose. This is why we call it deoxyribonucleic acid. Okay, we call this ribonucleic acid. We call this deoxynucleic acid. Deoxyribonucleic acid because there is oxygen eliminated in case of DNA. So again, the main difference between RNA and DNA, one main difference is the type of sugar. Ribose in case of RNA, deoxyribose in DNA. Purines are the same, adenine, guanine. A second difference is the pyrimidine. If I say type of pyrimidine, that will be a true statement. Why? Because in DNA, you have thymine, cytosine, okay? In RNA, you have uracil, cytosine. You don't have thymine or T in RNA. So purines here is the nitrogen base, which is basically either adenine or guanine. And those are the same. So the main difference will be the sugar. Okay, so this is a false statement here. Okay. Do you have any question for me so far?
No.
Okay, let's proceed here again. Here, this is very, I need from you to know this, because when I say the central dogma of molecular biology and the central dogma, this is how the information in the DNA is converted to phenotypes, to proteins. And we call the process or the flow of information from DNA to make proteins, we call this the central dogma of molecular biology. And there are two steps here. DNA, first of all, is going to be transcribed to give a messenger RNA, and then the messenger RNA is going to be translated to make proteins. As we are going to see this in more clearly in the last chapter. So when I say that the central dogma of molecular biology state that DNA is translated to RNA, no, DNA cannot translate. DNA is transcribed into RNA. And RNA is translated, not transcribed into protein. So the main problem here, it is the flow is right, DNA, RNA, protein, but the process is wrong. From DNA to RNA, we call this transcription. From RNA to protein, we call this translation. That's why the majority again got that one wrong. Almost 89% got it wrong.
Sorry, I missed that. What did you say the central dogma means? Central
dogma meaning the flow of information from DNA, okay, to make proteins. How proteins formed. So information is basically transmitted from DNA to RNA first through the process of transcription, and then from RNA to protein through the process of translation.
Thank you.
Yeah, this is important. You need to know in biology, central dogma, if anybody asks you what is this, you need to know to tell them this is the flow of information, how information is transmitted from DNA to phenotype as we can see it. How the information you carry in your DNA will be transformed into eye color or hair color or whatever. Okay, so this is the process here through transcription and translation.
Okay, thank you.
Okay. And yeah, this is another one here. And I can see roughly more than 50% of the class got that one wrong as well. So when I said during a laboratory experiment, we discovered an enzyme catalyzed a reaction, and that reaction had a delta G, change in free energy, which is minus 20 kilocalorie per mole. So this reaction is basically exergonic, okay, generate energy. So what if you double the amount of enzyme in this reaction? What you are going to expect the delta G will be? Again, this is a different rephrasing for a previous question, that enzymes has nothing to do with the delta G of the reaction. Always remember, Enzymes cannot change delta G of the reaction. A delta G is basically property of the reactants and products. Enzyme has nothing to do with this. The enzyme, what the enzyme can do is basically to facilitate the reaction, to lower activation energy needed for the reaction to start. So delta G is not going to change. So delta G is going to be remain the same, okay? No increase, no decrease in delta G. This is very, very important. Okay, make sense?
Yes.
Yes.
Okay, great. And yeah, this is, again, when we talk about this, when you try to do some kind of a fractionation or separation of homogenized cells, you have cells, you was able to squeeze those cells and get all the like cellular component out. You have the nuclei, you have the mitochondria. If it is a plant cells, you have chloroplast. Then you have cytoplasm, you have ribosomes. So if you do some sort of fractionation, okay, trying to separate those from each other. So when you increase the speed, so you need, when you require to greater speed of the centrifugation or separation, so it says which fraction here will be will require high speed of centrifugation to be able to get it, the smallest. So the smallest fraction, the smallest size will need lots of speed to get it precipitated. So if you compare those 4 different like structure in the cell organelles, you can see that the smallest is going to be ribosomes. Those are the smallest. Nuclei is big, it has lots, it has the DNA, so it's heavier, okay? Mitochondria, chloroplast, maybe they have the same kind of same mass, but ribosomes very, very tiny. So in order to be able to get this ribosome separated, you need to have lots of speed, lots of centrifugation force to be able to get it separated, okay? Yeah, here, say a newly discovered unicellular organism, isolated from this kind of environment. It has some features like a plasma membrane, flagella, peroxisome. So when you look at this, if you see an organism which is basically has peroxisome, it's an organelle. So now we are not talking about prokaryotes. You need to get prokaryotes away from the equation. So anything that say prokaryotes, you need to eliminate from this. So basically, we're going to eliminate number 1 and number 2 because this is a true eukaryotes. It has organelle. Prokaryotes, they don't have organelles. No nuclei, no organelles. Now, when you say, is it a motile, or non-motile? Again, flagella. Flagella, this is a motility organelle, basically. So when you see this, so that will be an eukaryote and motile. Okay? That makes sense to you?
I'm sorry, can you repeat that again? You said can you repeat that one more time?
Yeah, here, when you say peroxisome, it's an organelle. Prokaryotes don't have organelles first.
Yeah.
So you need to eliminate prokaryotes from the selection. That will
Okay.
leave only eukaryotes. So now, is it motile or non-motile? If they have flagella like that, so they are motile. So you're going to select motile eukaryotes.
Okay, so the flagella always is motile?
Yes, flagella, this is flagellated. Any cell that has flagella, it's like a structure that enables the cell to move from one place to another.
Okay.
Yes. Yes. Like
So,
bacteria, they have flagella, so they can move to different places. Yes.
So is the difference between motile and non-motile the flagella? So it's going to be movement, basically.
Yes. Yes.
Okay.
Yes. Yes. Okay. So that will leave only one. I need to go again here and do this exam and let me go to this one here. That question here. Okay. So if you look at this kind of, can you see this figure here?
Yes.
Okay, it's a very nice chemical or biochemical reaction, which is basically you are trying to see the correlation between the free energy and the reactant and products and the progress of reaction. So you have a reaction, and that reaction start with the reactants, which are A and B. So A and plus B are the reactants, and they are going to form product, which is C and D, whatever A, whatever B, whatever the letter is, but those are the reactant, those are the products. So now if you compare the free energy of the reactants and the products, this is the level of the free energy. So which one has more free energy, the reactants or the products? This is the products. If you extend the line, that will be the free energy of the products. And this is the reactant, this is the free energy of the reactant. So which one has more free energy available to do work?
Reactants.
Yes, reactant here, because it has, this is a scale, so they are high here, they have more energy than products. So when you have a reaction like that where the free energy of the reactants more, so that is going to be an exergonic. Exergonic because you have more energy in the reactant than the product. So in order for those to convert, they're going to release energy. And we call this delta G is going to be minus and exergonic. So now, here in this figure, you have the reaction going by two different things, via enzymes and via no enzyme reaction. Okay, so it's one of them with the enzyme and one with no enzyme involved. So for sure, we know that enzymes, this is when you have a reaction like that, so you need to put some energy first. We call this activation energy. So if you look here clearly, you can see that there is less energy needed or less activation energy needed than this one. So in this reaction, we call this enzyme based. So an enzyme lower the free energy, lower the energy needed to a reaction to happen. Okay. So if you look here, when I ask, first of all, is this reaction exergonic or endergonic? So if you look here again, going from high, like delta G, to lower delta G, the main difference between the delta G is going to be minus. So that is going to be an exergonic reaction here. Number two, which letter represent reactant products? Those are the reactants, and C and D are the product. Okay. Change in free energy. Change in free energy is basically the difference in free energy between reactant and products. So that will be, if you go here, okay, that will be D. So D is the difference in free energy between reactant and product. You don't need to measure from the activation energy of the reaction. You measure directly from free energy of the reactant and product. So the letter D here represent the delta G of the reaction, okay? And then it's a which letter represent activation energy required using enzyme. We know that enzymes lower the activation energy. So that will be lowering the energy here needed a little bit less than without enzyme. So B is going to be activation energy needed for the enzyme. C is going to be activation energy for non-enzymatic reaction. Okay, that was the answer for this question, and I'll be more than happy to answer any question you have for this particular one.
I'm just gonna find, I do have one question, I'm just gonna find it.
No problem, yeah, go ahead. Anyone had any question in this particular figure here?
No.
Okay. So yeah, what you said you have a question for this one or something different?
It's exam 2, it's just one we haven't talked about yet.
Yeah, go ahead. Yeah, tell me which one.
I'm just looking for it, sorry.
Yeah, that's okay.
It's 34. So it says, what is the major structural difference between starch and glycogen?
Yeah. Okay, let me see. Yeah. The main difference between starch and glycogen, they made of the same kind of sugar unit, and the main difference is going to be the branching of the molecule. So let me show you here. Share. So if you go here, okay, this starch. Can you see here? So if you look at starch, you can see all those are glucose, and they are linked together via a glycosidic, the kind of covalent bond that connect those glucose together. We call it glycosidic linkage. And the type of glycosidic linkage is the same. It is beta 1,4, okay? Beta 1,4 glycosidic linkage. What is the difference between starch and glycogen? Same building blocks, same kind of covalent bonding. The main difference is going to be the branching. If you look at glycogen, glycogen is more branching than structurally. They have more branch. See here, at each point, you'll see some glucose molecule coming out. There is another point of branching again. You will not see this in starch. So starch is starch made of amylose, and amylopectin. So the degree of branching is less, much less than glycogen. So to answer this, the main difference is the branching, how the molecule is branched, made of more complicated. Starch is more simpler than glycogen, okay? And now that will take me then, what is the cellulose? Cellulose also made of the same building blocks, which are glucose. But the main difference here, that cellulose made of the kind of linkage is beta 1,4 beta glycosidic linkage, okay?
Okay.
And it is mainly connected, it's like microfibrils made of those long polymers of sugar, and they are holding together and stabilized with hydrogen bonding. So you can't see any branching in the molecule in cellulose. So again, this is why I need from you to put more emphasis on this chapter. It's very important, because in the final, I'm going to select this chapter again, so you will see it again in your final.
Okay.
Great. So do you have any question for me so far? You have any question for me for either chapter 9 and chapter 10? Chapter 9 and chapter 10, cellular respiration and photosynthesis. Anything which is kind of confusing, or you need to have more, like to shed more light on it?
I just have a bit of a general question. So for chapter 9, we basically have to know the difference between fermentation, aerobic respiration, and anaerobic respiration.
Yeah, this is important, very important. You need first of all to know what is the purpose of cell respiration. It's very important to know that mainly any living cell go through the process of cell respiration is basically to generate energy. So all the energy which is kind of stored in the bond, like a potential energy, those energy need to be released. Okay, and when they release, they break down the molecules, and it will convert to ATP. And ATP is considered to be like the main energy source, where every single process in your cell recognize it and know exactly how to deal with it. Okay, and any enzymatic reaction, you cannot give the energy in the form of a molecule of glucose and ask the enzyme to take or extract the energy and use it to make something. This cannot happen. So the energy in glucose should be released in the form of ATP, and this is a molecule which is basically used for any reaction that needs energy. This is very, very important. So respiration, you need to know the difference between glycolysis. You need to know the difference between glycolysis, the Krebs cycle, and the electron transport, chemiosmosis, and ATP production in the electron transport chain. Very, very important to know those kind of information. Okay, I will not give you like, I will not ask you to tell me how to go from glucose to pyruvate. You know, I mean, structurally, I don't need from you to know any of the structure. Don't memorize any structure. I will not ask you the difference between glyceraldehyde 3-phosphate, for example, and the dihydroxyacetone phosphate. Okay, I will not ask you this, but you really need to know what is the main purpose of glycolysis. Okay, and what will be the end product of glycolysis and where it happens. Okay, and if it produces ATP, how many molecules, and is it going to be consuming ATP to produce ATP or what? Yes, they are consuming 2 molecules of ATP to produce 4 molecules. So the net will be 2 molecules of ATP generated. What is the end product? In glycolysis, glucose is going to basically split into 2 molecules of pyruvate. And the pyruvate will be involved in a Krebs cycle where it is going to go through further degradation to break down all this pyruvate to generate carbon dioxide, and more energy will be produced. So you need to know that the Krebs cycle is happening in the mitochondria. Where in the mitochondria? In the matrix of the mitochondria. Glycolysis is happening in the cytoplasm or cytosol of the cell. So this kind of information you need to be familiar. You need to know the difference between oxidation and reduction. Oxidation reaction, reduction reaction. Okay. Always oxidation, losing electrons. Reduction, taking or accepting or gaining electrons. Okay. You need to know the difference between NAD and NADH. Okay. Those are the electron carriers. And when it is oxidized, it is NAD. And when it is reduced, it will take electrons and it will transform to NADH. Okay. So if I ask you the difference between NAD and NADH, which one has more potential energy? Is it NAD plus or NADH?
NADH.
Perfect. It is one has more potential energy because this is the one that is going to give this energy via the electron transport chain and make ATP. Okay. This potential energy just associated with the hydrogen atom associated with it. So you have a hydrogen atom associated with NAD to create NADH, which has more potential energy than NAD plus. So you need to know this kind of information. How the process is regulated, ATP molecule structure of ATP is important. So if I give you like a short diagram like that, I need ask you to know those, say like the 6 different parts of that ATP is, you need to know this as well. So it's, I was preferring to put 9 and 10 separate, okay, and make 12, 14, and 15 and 17 together. But that will, I think that will be, so 2 in exam number 3 and 4 in exam number 4. So I decided to have like split equally. So you have 3 chapters exam 3 and 3 chapters exam 4. Okay, so I don't know if you prefer to have only 2 chapters for this exam and 4 for the next, or this is better to have it split between 2 tests. What's your opinion?
In my preference, I would say split, just obviously because we're on a time crunch, it's harder to learn 4 chapters at once than it is to better know 3, if that makes sense.
Yes,
but
yes.
That's what I feel.
Yeah, same.
Yes. I usually, when it was face-to-face, I always do like 2, 9 and 10 together, because I know that 9 and 10 are so connected. One of them is a catabolic reaction, breaking down, and the other one is anabolic reaction, making and building. And they are like opposite, you know, so I do those together. But this is for 4 months semester. This semester is so short, so it is, when it is online too, so it's kind of hard, I know that. This is why I said I'd like to split it better. So we have 3 chapters for this test, 3 chapters for the next test, and then I will make the final easy for you. I will select few chapters only for the final, and I will tell you exactly where to focus. I will not ask you to do all again. I will not. So I will select.
Thank you.
It will be too much to go through all this material, especially in a very short time. Yeah.
I feel like I understand chapter 10 because it is photosynthesis.
Yes.
But for the test, do you want us to tell you like the products that are made through photosynthesis and
Yeah, photosynthesis, you need to know, first of all, photosynthesis is an anabolic reaction. Anabolic meaning building and making. And when you talk about anabolic, that energy is needed, okay? So it is an endergonic reaction. And the source of energy is basically mainly for photoautotrophs. They use light energy or sun energy, you know, to make and build macromolecules from carbon dioxide. It's the first thing, the basics, and you need to know the equation that basically in photosynthesis, carbon dioxide is N, water is N, sunlight is N, and what comes out is going to be simple sugar like glucose. Oxygen is coming out as a byproduct, and energy is the most important thing. Sorry, not energy. It's not exergonic. So it will be carbon dioxide, it will be organic compound like glucose, it will be oxygen, okay? This is important to know this. Number 2, you need to know that in photosynthesis, there are the light reaction and the dark reaction. In plants, this happened in the chloroplast. So you need to have an idea about the structure of the chloroplast as an organelle. You need to have an idea about the pigments involved in the process. Those are the pigments that capture the sunlight, okay? And you need to know the difference between photosynthesis 1 and photosynthesis 2, okay? And the light reaction. The dark reaction, what is needed, you know, to make sugar, Kelvin cycle, how Kelvin cycle works, these kind of things. We need to focus more about carbon 4 and all those accessory at the end of the chapter. So I will mainly focus on the main point of that chapter, chapter 14, chapter 10. You know, I think that should be, let me show you here.
And I'm sorry, you said that exam 3 is going to be 9, 10, and 12?
Yes,
correct?
yes.
Okay.
Cell cycle, yes. Yeah. Yeah, you know, you are almost there. If you do good in this one, I think you'll be fine. I will again give you also some extra points for the chapter 3, just to give you, you know, like floating. If you do the extra credit for chapter 2, I will add those to your grade for test 2. And then we'll see how you are going to do in 2, 3, and this 9, 10, and 12. Yeah.
Thank you.
No problem. Have any
Yes,
question
thank you.
for me? No problem, guys. Thank you very much. Okay, great. So just be in touch with me if you don't mind. If you have any question, just, you know, email me, okay?
Okay, thank you.
Thanks. Good day.
Okay.
Thank you very much.
Okay, thank you very much. Bye-bye. Bye-bye.
Bye-bye.
Bye-bye.